This is our lecture that will cover skeletal muscle contraction, design of the spinal cord and brain, as well as neuromuscular junctions and reflexes. You will be held accountable for this material on your next lecture exam. If you need information clarified, you should come to your instructor for help. I'd like to start with a brief discussion on muscle function. Obviously, muscle allows us to move. Muscle also helps with our posture and our joint stability. Brief contractions and relaxations that look like shivers help produce heat, and facial muscles can help us communicate as effectively as the words we use. And lastly, muscles do control our body openings. The word muscle means little mouse. When you flex the muscle, the ball of muscle underneath your skin was thought to look like a little mouse. Some properties of muscle include being excitable. That means they can generate an electrical response. They are conductive. That means the electrical excitation can spread, in other words, travel. Next, it has contractility. That means it can shorten, and it has extensibility. In other words, you can stretch your muscle. Lastly, the muscle has elastic properties, and that basically means it can recoil back into its original shape. The prefixes myo and sarco mean muscle. I now want to talk about the three classes of muscle, and these three classes were generated based on histological appearance and function. Let's start with a brief overview. Skeletal muscle is striated. In other words, it looks like it has lines running through an individual muscle cell. Skeletal muscle is also voluntary. In other words, you have conscious control over your skeletal muscles. Well, at least most of them. Skeletal muscle is also multinucleated, and when we speak of an individual skeletal cell, we call it a myofiber. Cardiac muscle is also striated. In other words, it looks like there are lines running through the cell. It is involuntary. You do not really have conscious control over how your muscle beats. Next, the cardiac muscle appears branched, and I'll show you an example of that in a little while. And when we speak of cardiac cells, we speak of cardiocytes. Smooth muscle is non-striated and is involuntary and is fusiform in shape. In other words, it looks like a spindle. Let's elaborate a little bit more on cardiac muscle. Shown in the top left picture is a histological preparation of cardiac muscle. You'll notice that the cardiac cells appear branched, much like the freeway system in Orange County. The blue arrow is pointing to a special junction in between individual cardiocytes. That is called an intercalated disc, and we will talk about its important properties in another unit. You'll also notice the big purple ball-like structures. Those are the nuclei of each individual cardiocyte. Lastly, I want you to notice that the individual cardiocyte appears to have lines running through it. In other words, striations. Those striations occur due to the arrangement of myofilaments. I'll be talking about the myofilaments, actin and myosin, in a little while. I'd now like to talk about smooth muscle, just briefly. Smooth muscle is considered involuntary muscle. And if I asked you where you would find it in the body, I'm sure the intestinal tract would come to mind most quickly. Smooth muscle is fusiform in shape, and if you look at the cartoon drawing in the bottom right picture, you'll also notice that they are mononucleated. That means they have one nucleus. You'll also notice that they don't appear to have lines running through the individual muscle cells. In other words, in a histological preparation, we say they are non-striated. Now, does that mean that they lack contractile fibers? Do they lack myofilaments? No. Look at the top right cartoon drawing now. You'll notice that they still have their contractile fibers, but these myofilaments are not arranged in an organized pattern, and therefore, in a histological preparation, we do not see organized striations. Lastly, I'd like to briefly highlight skeletal muscle. You'll notice that in this histological preparation there appear to be lines or striations running through the skeletal muscle cell or myofiber. These striations are generated due to the organization of myofilaments, and I'll talk about the thick and thin myofilaments in a little while. Next, you'll notice that an individual skeletal muscle cell, or myofiber, has many nuclei found along the perimeter of the fiber. In other words, skeletal muscle is multinucleated. Lastly, skeletal muscle is considered voluntary. In other words, you have conscious control over whether it contracts or relaxes. Most of the skeletal muscle is voluntary. However, there are times that you will learn throughout the class when a skeletal muscle can be considered to be involuntary, like the diaphragm. An origin is an attachment of a muscle that remains relatively fixed during muscle contraction. An insertion is the movable part or attachment of a muscle. When a muscle contracts, the insertion moves towards the origin. The thickest part of a muscle is considered its belly. Actions come in a variety of terms such as flexion, extension, opposition, abduction, adduction, and the muscle that is primarily responsible for causing a particular action is called the prime mover. A muscle that helps with that movement is called the synergist, and a muscle that opposes that motion is called an antagonist. For example, for flexion of the forearm, you think of your biceps brachii, but if you want to extend your forearm, the antagonistic muscle would be your triceps brachii. A fixator is a muscle that helps stabilize a joint. It's now time for me to tell you about levels of muscle structure, and I have a cigarette carton analogy. It's not the healthiest analogy, I understand that, but it works nonetheless. Pretend you go to the store and you buy a carton of cigarettes. You bring that carton home and open the outer cardboard box. Inside you see individual cigarette packs. Now, take out a cigarette pack, unwrap the cellophane wrapper, and open the cigarette pack and pull out an individual cigarette. Look at that cigarette and notice that it is lined with paper. If you cut through the paper inside the cigarette, you would find shreds of tobacco. Well, that analogy actually carries over quite nicely to the levels of muscle structure. If we think of a whole muscle as our cigarette carton, then the lining of our whole muscle is our epimysium. In other words, that would be the cardboard box. Now, if we cut through the epimysium to pull out an individual cigarette pack, well, in our muscle, we would essentially be pulling out a fascicle. A fascicle is a collection of individual muscle cells, and much like an individual cigarette pack being lined with cellophane wrapper, a fascicle is wrapped by perimysium. Now, take out an individual cigarette. In our muscle, this would be like taking out an individual muscle fiber cell. If we could cut through this individual muscle fiber cell, inside we would find clusters of fibrils. This is much like our cigarette and cutting through the paper and seeing shreds of tobacco inside. An individual muscle fiber cell is wrapped by its sarcolemma. That basically means its cell membrane, but also a layer of connective tissue called endomysium. Now let's review and do this again. In this picture, starting at the top right, we see a person's arm with a deltoid muscle. Again, think of that as a carton of cigarettes. If we take out an individual pack of cigarettes from our carton, that would be much like taking out or looking at a muscle fascicle. And a fascicle is an individual group of muscle cells. Now let's take out of our fascicle an individual muscle fiber. Again, going back to our analogy, that would be like taking out an individual cigarette from the cigarette pack. If we could open up the muscle fiber and analyze the fibrils inside, going to our analogy, that's like opening up an individual cigarette and looking at the tobacco inside. So if we're looking at the myofibrils inside a muscle fiber, you'll notice according to this picture, that there are striations, or a lined appearance. And I'm going to talk about these myofibrils in more detail in the next few slides. You're going to learn that these myofibrils have this appearance due to the arrangement of myofilaments. Nuclei along the length of the muscle fiber come from satellite cells during the formation of the fiber. Now it's time for us to go from a large muscle down to a fascicle, which is a collection of individual muscle cells wrapped with perimysium. And in this black and white picture, we're looking at an individual muscle cell or myofiber. If we peel away the sarcolemma of this myofiber, you'll see in the cytoplasm of this muscle cell, we see many repeating structures called myofibrils. Let's do this again. Sarco means muscle, and lemma means plasma membrane. So, sarcolemma is the cell membrane around the muscle cell. Sarcoplasm means the cytoplasm within a muscle cell. In the cytoplasm, you'll notice that we have these repeating rod-like structures called fibrils. Next, I want you to see these yellow structures shown here in this cartoon. These are called T-tubules or transverse tubules. These are invaginations of the sarcolemma. Please note, this is not to say that the sarcolemma has holes punched through it. No, the integrity of the sarcolemma is still maintained. These are not rips nor tears, they are merely extensions of the sarcolemma that invaginate into the cytoplasm, and you'll notice the sarcolemma wraps around each individual myofibril. Why might these T-tubules be very important? Well, you're going to learn pretty soon that these T-tubules are going to allow an electrical current to travel deep throughout the cytoplasm and basically engage every single myofibril. We'll talk more about that later. Next, I want you to notice that this blue web-like structure is called the sarcoplasmic reticulum. I'm sure that reminds you of an organelle called the endoplasmic reticulum. Well, sarcoplasmic reticulum is basically the same thing, except inside a muscle cell. In addition to helping with protein synthesis, the sarcoplasmic reticulum also stores a tremendous amount of calcium within it. And you should already start to appreciate that when calcium is released from this organelle, a chain of events will happen that will allow a muscle fiber to contract or shorten. Next, I want you to notice that the sarcoplasmic reticulum has these swollen endings. In other words, areas where the sarcoplasmic reticulum looks larger. And you'll also notice that these terminal cisternae occur on both ends of a T-tubule. So terminal cisternae are the swollen ends of the sarcoplasmic reticulum that are in functional contact with a T-tubule. In fact, this functional contact is very important for the transmittance of the electrical signal. And I want you to understand that the terminal cisternae on either end of a T-tubule is called a triad. Triad meaning three. Think of it as three T's next to each other. Terminal cisternae, T-tubule, and on the other side, another terminal cisternae. Okay, we have gone from a large muscle down to a fascicle. Remember, a fascicle is a collection of individual muscle cells or fibers surrounded by perimysium. If we now take an individual muscle fiber or cell and peel away the sarcolemma, as demonstrated here in the top left picture, underneath the sarcolemma you'll notice that we see many dark black nuclei. Again, skeletal muscle is multinucleated. Inside the cytoplasm, in addition to the nuclei, we see rods of myofibrils. The center portion of this picture shows an individual myofibril expanded for view. You'll notice terms like Z-disc and H-zone, A-band and I-band. I'll explain what those mean in a little bit. From one Z-disc to another Z-disc, this region is called a sarcomere. and a sarcomere is the individual unit of the muscle cell. In other words, it's the functional unit. This is where contraction is going to occur. Now, I want you to notice that we have bands of thick filaments. And these thick filaments are shown as bright red structures. We also have structures that are called thin filaments, and they are shown here as the light blue structures. You'll notice the thin filaments are anchored to the Z disc. I'm going to explain what these thick and thin filaments are doing in the cell in the next few slides, but right now I would like to talk to you about their overall arrangement. I want you to pretend that you are in your lab classroom, and I want you to pretend that you are floating up by the ceiling and looking down at the tables and at the chairs staggered in between your lab tables. What if there was a giant light shining through the floor of our lab classroom? Do you agree that the tables in the classroom would obstruct the light from reaching your eye, and this would create a shadow? Well, the thick filaments in a sarcomere, shown as the red structures in the bottom picture, do much the same thing when a fibril is viewed under a microscope. These thick filaments obstruct the light from getting through, and so it creates a shadow-like appearance. Now, the thick filaments create what's called the A band. Notice that that term is shown here in the middle picture. How can you remember that the thick filaments create the A band, which basically is like a shadow when looked at under a microscope? Well, what is the second letter in the word dark? That's right, A. So you'll now remember that the thick filaments create the dark band, and this is known as the A band. Now I want you to think about the individual laboratory chairs that are staggered on either side of our lab tables. If you look at the bottom picture and notice the helical or intertwined light blue lines, the thin filaments, they too are staggered in between the thick filaments. So if you were looking at the light shining through our lab floor, the spaces where our desk chairs are found would allow more light to shine through. Now, how are you going to remember that the thin filaments, where they attach to the Z disc and don't have any overlap with the thick filaments, constitutes the I band shown in the middle picture? What is the second letter in the word light? That's right, I. So where we have thin filaments only, with no overlap with thick filaments, a lot of light would be able to shine through. So that's how I want you to remember the I band. Now let me go a step further. Let's pretend we're all seated in our lab classroom. You are all seated in your chairs and you're writing your notes on your lab table. Let's pretend I asked the whole class to slide over to the middle of the classroom. What would be the easiest thing to do? Move the tables to the center of the classroom? No, they are anchored in place. Rather, you all would slide on your wheeled chairs to the center of the classroom. And that is exactly what the thin filaments are going to do during muscle contraction. The thin filaments are going to glide across the thick filaments. In other words, the A band does not change in length. Rather, the I bands will shorten and even disappear when a muscle is fully contracted. We will go through this process again and again and again. I now want to spend more time on myofilaments. It is the arrangement of myofilaments within a myofibril, within a myofiber, that causes the myofiber to look striated. Let's start with the thick filament. Thick filaments are composed of many individual myosin molecules. If you look at the bottom picture, a myosin molecule looks like a mutated golf club. In other words, it looks like it has a long rod with a single club head, but has mutated to create another club head. So I like to think of this myosin molecule as a double-headed golf club. On the head of this myosin molecule is a unique enzyme called ATPase. ATPase, the name tells you what it's going to do. It is an enzyme that will cleave ATP, or adenosine triphosphate. And when it cleaves ATP, it will release an inorganic phosphate group and ADP, or adenosine diphosphate. Through this action, a tremendous amount of energy will be released, and it is this energy that is going to allow the myosin heads to ratchet or pull the thin filaments across them. Now I want to focus on thin filaments, shown as the light blue intertwined strands. Thin filaments actually are created by multiple different molecules. I'm going to talk about actin, I'm going to talk about troponin, and I'm going to talk about tropomyosin. All three of these molecules will create a thin filament. If you only have one of them, you do not have a full, complete thin filament. You need actin intertwined, you need troponin, and you need tropomyosin. You're going to learn that tropomyosin and troponin act as regulatory units. In other words, they are going to cover the actin filaments so that the myosin heads cannot bind or engage with them. If the troponin and tropomyosin regulatory units are removed, this will expose actin, and then the myosin head will be able to bind with actin and slide the thin filaments toward the center of the sarcomere. That, in a nutshell, covers contraction. I now want to continue our discussion on the sarcomere. The far right picture shows one individual sarcomere bounded by two Z discs. And you'll notice that there are thick and thin filaments interdigitated between each other. I'd like to talk first about the thin filaments. Shown at the end of this arrow are globular subunits of actin protein, and these globular subunits can link together to form a filamentous actin molecule, shown here at the end of this arrow. You'll notice that it's a double-stranded intertwined filamentous actin molecule. Even with this long F-actin filament, we still do not have all the thin filaments put together. Additionally, we need troponin and tropomyosin. Troponin is a protein that binds both actin and tropomyosin. Additionally, it binds to calcium, and this will be an important part of muscle contraction, which I'll discuss later. Tropomyosin covers the active sites of actin, the filamentous actin. Then this prevents the interaction with the myosin heads. I now want to talk about myosin. Here is an individual myosin molecule at the end of this arrow. And when we have a lot of myosin molecules interacting with each other, with their heads radiating out, then we'll have a myosin filament. And I'll show you this in a little bit. The second arrow is showing an individual myosin molecule. You'll notice it's made of two heavy chains and two light chains. The head of the myosin molecule contains an ATPase enzyme. Basically, this means that it will allow ATP to be cleaved, and the result will be that the energy released will allow the myosin molecules to ratchet their heads back and forth. I'll talk more about this later. You'll now notice this third arrow. When we have many myosin molecules associated together with their heads radiating outwards in all directions, now we have a thick filament. I have taken you through the gross anatomy of a muscle. We have gone from the whole muscle itself down to collections of muscle fibers called fascicles, to the individual muscle cell called a fiber, to the long strands of proteins called fibrils, down to the myofilaments that compose a sarcomere. Now I want to back up and show you this picture in the middle of the slide. You'll notice that there is a transverse section through the spinal cord, and leaving the spinal cord are two individual motor neurons. Neurons that innervate muscle are called motor neurons. You'll notice that these two motor neurons are innervating individual muscle cells that belong to a larger muscle. A motor unit is a collection of muscle fibers innervated by a single motor neuron, and the muscle fibers within this motor unit are generally of the same type, that is, either fast or slow, and I'll be discussing motor fiber types in a little bit. motor units overlap, which provides coordination, and we need recruitment of motor units in order to generate a strong muscle contraction. In other words, if you were to pick up a piece of paper using your biceps brachii to flex your forearm to pick up that piece of paper, you would likely only use a few motor units in order to stimulate contraction in a few muscle cells. But if you needed to pick up 10 bricks, let's say, you would need to recruit many more motor units in the biceps brachii muscle. I want to reiterate that a single muscle cell has only one neuron communicating to it, but a single neuron can innervate many fibers. How can you think about this? Well, let's pretend that I am a single neuron and I am communicating to you all, my class. You are individual muscle cells. You have only one teacher speaking to you, communicating to you, but I communicate to many individual muscle cells, and that is basically a motor unit. Shown again, let's pretend that these green and blue muscle fibers compose a whole entire muscle. We would expect more than one motor unit. In fact, shown here is a single motor neuron communicating with 3 individual muscle fibers. A second motor neuron communicates with the remaining muscle fibers. If we wanted only a gentle contraction, probably only the first neuron would stimulate those first 3 muscle fibers. But if we needed a very strong contraction, then both motor units would likely stimulate their muscle fibers to generate a strong muscle contraction. I now would like to talk about the motor neuron itself. This picture represents a single neuronal cell that innervates muscle. In other words, it is a motor neuron. On the left side of the picture, you see the soma, or the cell body. You'll notice in the soma, or cell body, that there is a filled-in blue circle, and this represents the nucleus, where gene transcription occurs. You'll also notice that next to the nucleus are several oval-shaped structures. These represent the endoplasmic reticulum and Golgi apparatus. You'll also notice several circle shapes emerging from the Golgi apparatus. This represents the synaptic vesicles. On the right side of the picture, you see the terminal bouton, also sometimes called the synaptic terminus, filled with many vesicles, and the red dots represent acetylcholine. Now, let me tell you what happens. Synaptic vesicles bud off of the Golgi apparatus and transfer down to the terminal bouton by axoplasm streaming. Once in the terminal bouton, acetylcholine molecules, this is a type of neurotransmitter, are packed within the synaptic vesicles. When an appropriate signal comes through the neuron, the synaptic vesicles will actually fuse with the terminal bouton membrane, shown right now. Once this fusion occurs, the acetylcholine can now diffuse into the synaptic cleft. The acetylcholine is the chemical trigger that can bind to a receptor on a downstream cell.